So this micrograph here shows the steps in the initial steps of the receptor mediated endocytosis of low density lipoproteins, LDL, and these are electron micrographs. And so here in this image, the LDL particles are labeled with a protein called ferritin, and that's so that we could see it through the electron microscope. And so you see here in this image, all the dark spots here are the electron dense regions of the LDL coated proteins. And so this was an experiment done in which cultured human fibroblasts were incubated in a medium containing LDL particles that are covalently linked to the electron dense iron containing protein ferritin. And so the cells were incubated at 4 degrees Celsius in a solution of LDL. And so what this does is basically it allows the LDL to bind to all the receptors on the cell surface. But endocytosis does not occur until we raise the temperature to 37 degrees Celsius. At which point, now that all the receptors on the cell membrane are bound to the LDL particle, raising it to 37 degrees Celsius, now the endocytosis proceeds, and so you see this formation of clathrin coated pit here, and you see all the LDL ferritin labeled LDL are right there on the pit area. And so over time this pit will become a budding vesicle, which will be pinched off by the dynamin protein. And once the budding occurs, the vesicle transporting the ferritin tagged LDL will make its way into the endosome, shown here in this micrograph here. And so LDL is this protein complex, which is basically this spherical particle lipoprotein. And so it has an outer polar surface composed of phospholipids embedded with unesterified cholesterol. And in the interior we have a core of cholesterol ester, and these are hydrophobic, and sometimes other molecules are embedded in the interior, including triglycerides along with the cholesterol esters, as well as some vitamins. And the protein that's wrapping itself around this spherical particle is called apolipoprotein B, and this allows the LDL to bind to its receptor. And so from that experiment up there, where they incubated the cells at 4 degrees Celsius and exposed them to the LDL solution until all the receptors on the cell membrane were bound to LDL. This shows the populations of the different LDL after raising the temperature to 37 degrees Celsius. And so you see that initially all the LDL were in the bound state, but very shortly after temperature is raised, most of the LDL is internalized. And about 15 to 20 minutes into the experiment, the degradation of the LDL in the lysosome proceeds, and so you have this upward trend in degradation and this rapid decline in LDL that is bound, and also a big decline in internalized LDL en route to the lysosome. And so here's the summary of the whole process of the LDL transport into the lysosome. So you have this LDL particle in the bloodstream. You have these LDL receptors on the cell membrane, and they use this special arm here composed of 7 cysteine residues to bind to the ApoB protein that wraps itself around the LDL particle. Once contact is made here and the receptor is bound to LDL particles, this cytosolic segment of the receptor makes contact with a AP2 complex protein right underneath in this coated pit. And once this interaction here happens between the cytosolic receptor protein and the AP2 complex, the formation of the vesicle will happen, in which dynamin will catalyze the pinching of the clathrin-coated vesicle, and then afterward the shell, this protein shell with clathrin, will disassemble, and you have now this early endosome vesicle transporting the LDL with its receptor. Once it makes its way into the late endosome, the pH of the late endosome is lower than it was in the plasma membrane, and so this would favor the dissociation of the LDL particle from its receptor, and the receptor would be transported back to the plasma membrane, and then afterward the late endosome would fuse to the lysosome, where the LDL particle is processed into amino acids, cholesterol, and fatty acids. Now the sorting signal on the LDL receptor is this one-letter amino acid code MPXY. You also have this segment here called the beta propeller domain, and also this ligand binding arm in pink, which is 7 residues long, and that binds directly to the ApoB protein on the LDL. And this is at pH 7 where binding occurs. However, when the bound receptor and ligand get to the endosome, where pH is lower at pH of 5, the LDL will dissociate. It will basically be released from the 7 residue arm here, and the surface of the beta propeller here in green becomes positively charged, which would cause it to bind to this negatively charged cysteine residue arm. And here's a electron density map of this binding. There's also a similar process that occurs with the transport of iron into cells, and this occurs through ferrotransferrin protein, which carries 2 Fe3 plus iron ions. They bind to a plasma membrane receptor of transferrin, and once this interaction is made, the transferrin receptor will find a coated pit, and that will then proceed with the endocytosis, and dynamin will cause the pinching of this vesicle. Then the clathrin and AP2 complex is shed, and once inside the late endosome, you can see here that the ferrotransferrin will release the iron ions into the late endosome, and the iron ions become reduced from Fe3+ to Fe2+. And now that the protein doesn't have its iron ions, it is known as apotransferrin, which is recycled back to the cell surface, shown here. And once at the cell surface, the receptor will dissociate from its protein.